The University of KwaZulu-Natal's College of Agriculture, Engineering and Science is committed to making a difference in our world, from the classroom and laboratory to the field. It provides one of the finest study and research environments on the African continent and is recognized internationally as a center of excellence. Agriculture, Engineering and Science is one of 4 colleges that make up the University of KwaZulu-Natal and is home to over 8,700 students and approximately 700 staff. 3 of the university's 5 campuses form the core of agriculture, engineering and science activities in the region. The college comprises 5 schools: Agricultural, Earth and Environmental Sciences, Engineering, Chemistry and Physics, Life Sciences and Mathematics, Statistics and Computer Science. Engineering is offered on the Howard College campus, Agriculture on the Pietermaritzburg, and all the other sciences on both the Pietermaritzburg and Westville campuses. Exceptions are Agricultural Engineering, which is offered in Pietermaritzburg, and Geology, which is offered at Westville only. To compete with the best and to meet the challenges of the 21st century, South Africa needs highly skilled professionals in the areas of agriculture, engineering and science. We offer a wide array of interesting and innovative degree programs that open doors to a variety of different careers. Some degrees are unique to UKZN and are not offered at other tertiary institutions. For example, we are the only university in South Africa to offer a fully accredited agricultural engineering degree and one of only 2 universities in the country to offer a BSc in land surveying. Our postgraduate food security training program is the only one in the world, and we have the widest range of agricultural disciplines at any single South African or African campus. The choices of degree and the options on offer may seem confusing at first. Engineering degrees are more specific, and the options are usually career-focused. Emphasis is placed on training in the specific principles which engineers deal with in their professions. Engineering offers 7 different professional specializations. There are also degrees in land surveying or geomatics and property development. In science, there are structured or career-focused degrees. These include fields as varied as chemical technology, marine biology, industrial and applied biotechnology, and computer science and information technology, to mention a few. There are also more general degrees with a lot of flexibility. In this case, students have the choice of studying a BSc M stream in maths, physics, computer science, and statistics. All of these have a strong mathematical component. The other option is a BSc LES stream with a focus on the life and environmental sciences with subjects such as geography, genetics, botany, hydrology, zoology, ecology, and microbiology. In agriculture, there are various different options that involve practical and scientific applications. Students can combine science subjects with specific agricultural disciplines for a 4-year Bachelor of Science in Agriculture degree. These are more career-orientated. Agriculture and agricultural management are 3-year degrees with a higher practical component. Dietetics is a professional qualification that deals with the role nutrition plays in the promotion of health and the treatment of disease. Most degree programs across the college have field trips and projects that contribute significantly to the curriculum. This ensures that students learn in real-life situations under full field conditions. Many of our lecturers have received Excellence in Teaching awards, are recognized internationally, and are rich in practical experience. Students have access to excellent facilities that rank amongst the best in the world. 300 million rands has recently been invested in new teaching and research facilities, state-of-the-art laboratories, and improved equipment. The 30 million rands new School of Engineering building is the envy of engineering campuses across the country. Agricultural students have access to a 356-hectare research and training farm situated near the Pietermaritzburg campus. This facility provides students with hands-on training and is used extensively as an outdoor laboratory for teaching and research in areas such as animal and poultry science, horticultural science, and agricultural engineering. Adjoining Ukulinga is the Bisley Valley Nature Reserve, which supports a diversity of plant and bird species, as well as some large mammals. It provides a rich learning and research environment for staff and students. Based on the Westville campus, the college possesses unique facilities for research into higher voltage power transmission. These facilities are available in only 3 other places in the world and form part of the ESCOM-sponsored Science and Technology Innovation Park. The Science and Technology Education Center showcases the college's scientific achievements and promotes the public understanding of science, engineering, and technology within the region. The college houses a wide range of sophisticated equipment in its microscopy and microanalysis units, staffed by highly qualified and skilled technical specialists. The College of Agriculture, Engineering, and Science prides itself on its graduates who are highly sought after. In a market characterized by a shortage of qualified professionals in science and technology, our graduates are readily employed around the world in the fields of agriculture, science, commerce, industry, engineering, and research, to name just a few. Research plays a major role within the college and operates at the cutting edge of technology. College has the highest research output in the university and produces the most research publications. There are over 60 well-established research groups and centers within the college. The African Center for Crop Improvement caters for students from all over Africa. It is one of only 2 African plant breeding programs on the continent. Based in the School of Agricultural, Earth and Environmental Sciences, the African Center for Food Security focuses on alleviating hunger in Africa. It offers food security training programs, undertakes academic and applied research, and provides policy and program support. Promoting sustainable engineering practice is the Center for Research in Environmental, Coastal and Hydrological Engineering, or CRESH. It deals with the interaction between engineering and the natural environment. The Mechatronics and Robotics Research Group in the School of Engineering provides an excellent research and education environment for its senior undergraduate and postgraduate students. Judged one of the best research centers at the university, the Center for Plant Growth and Development focuses on high-quality research in the plant-based disciplines of plant physiology, plant molecular biology, and ethnobotany. The Center for Quantum Technology deals with physical phenomena at the microscopic level. Quantum physics led to the development of the transistor and the laser and is currently being used to develop quantum computers and quantum cryptography. The college is committed to access and readdressing the disadvantages and inequities of the past. The highly successful Unite Engineering program and Science Access Foundation and augmented programs offer alternative access to university, equipping students from disadvantaged backgrounds with the skills and knowledge to pursue their degrees of choice. Both these programs contribute significantly to advancing Black African scientists and engineers in South Africa. Staff and students contribute to a number of outreach and community initiatives that focus on social responsibility, sustainable development and upliftment of the wider community. The college also engages in activities involving the pursuit and understanding of science, engineering and technology through exhibitions, festivals and roadshows that target high school learners and the general public. The college has minimum entry requirements for its programs. These differ from degree to degree, but all require a national senior certificate and minimum performance levels in mathematics and either physical or life or agricultural sciences. For those who do not meet the minimum requirements, access programs are available. Additional information and brochures can be obtained from the contact information accompanying this video. The College of Agriculture, Engineering and Science is proud of its contribution to the university, the region and the country.
UKZN is one of the best. I definitely understand it's one of the best in South Africa. So for me, there was no other institute that I would have preferred other than UKZN.
We have the best facilities here in terms of laboratories, in terms of equipment, in terms of human resources, in terms of literature.
UKZN is one of the best universities in the country that teach agriculture or natural sciences.
What I think is good about this college is the level of international collaboration that we have. We have friendships with universities all over the world, which means that we're keeping up to a great international standard.
UKZN has got much more facilities and it's recommended worldwide to do your science studies here. Life begins here. Yes.
We continually strive to be academically excellent, innovative in research, and critically engaged with society. We invite you to join us or work with us to be part of this exciting team.
Good evening, students, and welcome to our second session of the Matric Helpline. If I can ask you all to have your mics on mute for me, please. And we will begin shortly. I know quite a few students from session one did express interest to join tonight, so I'll just give people 2 more minutes. It's 5:58, so they can join. In the meantime, I'll introduce you to my colleague, Dr. Minisha Singh. She will be addressing your concerns or questions on the chat of this Zoom meeting. And you can, as I said last time, put all your questions up there on the chat and she can answer them. Or if you need or have a question based on what I'm going to be teaching you tonight, you can also put it on the chat and Dr. Singh will relay that message to me. Dr. Singh will also be posting a few links for you to click on to register and also to find previous videos that we've uploaded for the Matric Helpline. So please make sure that you actually read the chat and see the things that she's going to be posting up there for you guys. Okay, let me just get my PowerPoint ready and we'll begin in 1 minute. Okay, students, can you see my screen there? You should be seeing a PowerPoint slide. Just gonna put it on slideshow.
The mouse on the side.
Sorry, it's just taking a while. Okay. Okay, can everybody see that? You can just shout out. Can you see, it should say Matric Helpline 2022, session 2.
Yes, I can see it.
Oh, thanks, Nams. Okay. Okay, so let's begin. Today we're going to go through your next 2 topics of human responses to the environment and homeostasis and the endocrine system. Okay. Sorry, I don't know why this is not working. Do you see the next slide?
Not as yet.
Okay, just a moment. I'm very sorry. There seems to be a technical error. It's not changing my slide.
Go.
Okay, can you see my screen yet?
Yes, can see the whole PowerPoint app.
Can you see my slides on the left?
Yes.
Okay. Okay, there you go. It's working now. Okay, so we are, as I said, looking at human responses to the environment and the endocrine system and homeostasis. Okay, so a few things you need to know in these topics. Okay, so obviously the central nervous system includes the brain and the spinal cord. So you need to know structure and function of the brain and spinal cord. Okay, all the components, the parts of the brain and their functions. Okay, that is very important. You'll definitely be asked questions on that in terms of labeling. They'll give you a diagram and you'll be asked to label it or state the function of the parts. Okay, then you have the peripheral nervous system. So you need to know the autonomic and somatic nervous systems. Okay, so autonomic means, think of the word auto, it happens automatically. So that is your involuntary system. Okay, and the somatic system means that it's got to do with your somatic cells or your muscles, that type of thing. So it is talking about responses. Okay, so a voluntary movement. Okay. Okay, then your sense organs like the eye. Okay, you'll need to know the following subtopics in under the eye: accommodation of the eye, pupillary mechanisms like when the pupil changes based on light, and then visual defects. Okay, like people who have farsightedness, etc. Okay, and then the ear, you need to know the structure and function of all the parts of the ear. Okay, and also I think it's come up in a past paper about dealing with the a disease of the ear, okay, or an illness of the ear. Okay, and then in the endocrine and system and homeostasis, you need to know all the glands, the hormones that they secrete, and the role of that hormone. Okay, this is very important. And then you need to know some disorders linking up to the endocrine system, such as diabetes, etc. Okay, and then negative feedback systems. This is under homeostasis. Okay, so you need to know how the body adapts to different conditions. Okay, like if it's too hot or if it's too cold, or if there's too much salt in your body, if there's too much sugar in your bloodstream. Okay, you need to know these negative feedback systems. And then the organ that deals with homeostasis is the skin. Okay. So those are the subtopics that you must be confident with. Okay. Okay, so let's start with some easy marks. Okay, give the biological term 4. Okay, you're going to just shout out like we did the other day. Okay, you can just put it in the chat or shout out the answer. Okay, bundles of nerves connecting 2 hemispheres of the brain. What is that? What's the term for that? Anybody? Yes, I can hear somebody thinking. What's the bundle of nerves connecting 2 hemispheres of the brain called?
Don says corpus callosum.
Yes, that is correct. Okay. Then what's the covering of fatty material that speeds up a nerve impulse in a neuron? Any takers? Okay, these are things you need to know, guys. So everybody
Myelin
should
sheath.
know that. Sorry, yes?
The myelin sheath.
Yes, that is correct. Okay, what reflects light and focuses the image onto the retina?
Don says the cornea.
No, try again.
The lens.
Yes, that is correct. Okay. So the lens is what is going to refract the light or bend it and focus that light onto the retina. Okay, and what is the term for bones that are responsible for transmitting and amplifying sounds in the ear? I'll give you a clue, it's 3 bones.
Don says ossicles.
Yes, that's correct. Okay, then a gland that discharges its secretion through a duct into the body cavity.
Exocrine glands.
Exocrine glands, very good. Okay, so you must know that an exocrine gland secretes its or discharges its secretion through a duct into the body, whereas an endocrine gland is ductless. So that now produces hormones and releases them into the bloodstream. Okay, so there's no duct for endocrine glands, but exocrine glands have ducts. Very good. Okay, if everybody can just take a quick look at the chat. Dr. Singh has posted a register link for you to click on, and you can register yourself there. We're compiling a database, so you will receive notifications from us for further helpline news. like when we might have the next one, or if you want to access videos and things, you can also click on the website link that Dr. Singh has posted. Okay, if you click on the website, you'll be able to find last year's videos. Okay, so it is up there for you. Okay, so let's begin with the short answer questions now. Okay, I hope everybody has their pen, paper ready to write down. Okay, that's the only way you're going to learn, if you're going to actually jot down these answers. And also practice answering them for yourself. Okay, don't just wait for me to give you the answer. Try and answer it yourself as well. Okay, and it's frozen again. So sorry. Okay, can you see that now?
Yes, you can see your slides on the left.
Okay. Okay, so you've been given a diagram 1 and diagram 2. Okay, and it is asking you for the labels of part C. Okay, and C is this gray looking bit in the middle here, or it's just like slightly dotted, that part in the middle. Okay, but what is this entire section? Okay, if that would give you a clue to what C is. Okay, and the answer is the spinal cord, but if you're more specific, you would say the gray matter of the spinal cord, because it's showing you the inside there. You can see a white section around it. Okay, that it would be the white matter. Okay, then what is D? D is very obvious, it's the cerebrum. Okay, next is give the answer, give the letter and the name of the part in diagram 2 that receives impulses from rods and cones. That would be the cerebrum. So what letter is that? Hello?
No answers yet.
Anybody, you can just shout out and answer me.
Don and Nandi says D.
Correct. Okay. Receptors in the heart muscles. Which part of the diagrams
F.
receives impulses from receptors in the heart? It would be F, which is the medulla oblongata. Okay. Then name the process taking place in diagram 1, which minimizes injury to the body. What is this whole diagram showing you? It's showing you a reflex arc, okay, which is performing a reflex action. And why would that minimize injury to the body? What type of action is this? Students? What kind of action is a reflex action?
Nandi says
It's a very fast action. Yes, very good. Okay. And that would minimize injury to the body because it's gonna save you from hurting yourself. It'll save you very quickly from hurting yourself because you have a reflex. And it happens very fast because these impulses are only being transmitted to the spinal cord. It's not traveling all the way to the brain, waiting for the brain to give a response to this effect. It's going straight from the sensory organ to the spinal cord, and the spinal cord is giving a response. It's giving, it's telling the effector muscle what to do. Okay, so it's a very fast action. Okay, give the letter and the name of the neuron that transports impulses from the central nervous system to the effectors. And that is your motor neuron. Okay, so that is what's now going to, for example, if you put your finger on a hot plate, your motor neuron will now receive the impulse from your spinal cord, and that motor neuron has its nerve plate ending in the muscle, and it will cause your finger to be pulled away from the hot stove. Okay. Okay, please let me know what you can see on the screen.
You can see your slides on the left.
Oh, okay. Okay, can you see a big version now?
Yes.
All right. Okay, so this graph, Nimisha, if you can just tell me what's on top of the graph there.
The top of the graph says changes in pupil diameter over time.
Okay, so you can see the size of the pupil is changing over time. Okay, so which structure in the human eye is responsible for the changes indicated in the graph? So which structure is responsible for changing the diameter of the pupil?
Nandi and Don says the iris.
Correct. Okay. And during which period of time was the person moving from dim light to bright light? So what happens when a person moves from dim light to bright light? What happens to that pupil?
Nandi says the pupil will get smaller.
Correct. Okay, so at which point of the graph does this happen?
(20 to 30 seconds).
Yes, that is correct. Okay, describe the changes that took place in the eye that led to the diameter change indicated from 30 to 35 seconds. So what's happening here from 30 to 35 seconds? Your eye is adjusting. Okay, so it's your pupil is adjusting to deal with this bright light. Okay.
Q says the circular muscles contract and the radial muscles relax.
And what does that do?
And Nandi says the suspensory ligaments are slackened.
Okay, so you're now in bright light. So yes, you're correct in saying that the circular muscles of the iris contract and the radial muscles relax. So what does that do to the pupil? It causes the pupil to decrease in size. Why does it decrease in size? Because it's trying to let in less light, because it's in bright light. Okay, so we're trying to limit the amount of light so the pupil gets smaller. Okay. Now draw a label diagram of the front view of the eye to show the exact size of the pupil at 20 seconds. Okay, so when the pupil is nice and wide. Okay, as indicated in the graph. Okay, so I'll just give you a few rules for biological drawings. Okay, you're just going to do a basic outline. Okay, you're not going to be shading anything in, you're not going to be coloring anything, you're not going to be drawing magnificent works of art. It's just going to be a basic line drawing of an eye. Okay, so it's going to be like an almond shaped eye with a circle in the middle. That's your actual iris, and then your pupil, and you're going to put in those radial muscles and those ciliary muscles, okay, just to show the size of the pupil. And then it's going to be a wide circle. Okay, so you can draw that, and then you must always, whether they ask you or not, and in this question they've asked you for a labeled diagram, so you're going to label the pupil, label the radial muscles, label the circular muscles. Okay, and you're going to also put a figure title at the bottom of your diagram. Okay, it always goes at the bottom. You're going to say figure 1, drawing of the front view of an eye to show size of the pupil in bright light, in dim light. Okay, is everybody with me? That's what's expected of you for a labeled diagram, a labeled biological drawing. Okay, so you must make sure that you have all of that detail. Okay, you don't have to spend time making it look amazing. It's not art, it's just biology. Okay, so make sure that you follow those rules. Okay, I'm just gonna ask everybody to mute themselves. I don't know who that was, but please do mute so that I can continue. Okay, give the letter of the name of the part that A controls one directional transmission of impulses. What letter is that?
Nandi says E.
What is E?
Now remember the question says letter and name. Q says nerve fiber.
Okay, so the question is a bit tricky. It's saying one directional transmission of impulses. Okay, which means that it can only go one way. Okay, so it's going from something to the next thing. It's not going back and forth. and that would be your synapse. Okay, that is where, and that is where transmission of impulses occurs. Okay, so we're looking for a synapse, and that would be at letter B. Okay, that's the answer to that one. It's quite, that one was a bit tricky. Okay, and then what transmits impulses from the sensory neuron to the correct motor neuron? So look at your diagram there. You can see A is going to B, and then you've got something in your gray matter there, and then you've got a line coming from E to F. So what's in the middle there?
What is C?
Sorry, yes, you can look at the other side for the letters. So that is C, and what is C? What is C?
Connector neuron. Mshlongo and Mfanelo says connector neuron.
Okay, or the interneuron. Okay, so connector neuron or an interneuron. Okay, then what transmits impulses to the cell body? That would be your dendrites. Okay, so dendrites transmit signals to the cell body, and an axon transmits signals away from the cell body. Okay, so where's our axon? Sorry, where's our dendrites? Which part is showing a dendrite? Where the impulse is coming from? In your diagram, you can see a person's finger getting burnt by a candle, the flame of a candle. So he's receiving an impulse of heat.
Mshlongo says A.
A is correct. Okay, so the dendrite there in that person's finger is now going to transmit that impulse of heat to B. That's the cell body there. Okay, give only the letter of the neuron that is damaged when a person is able to feel pain but cannot react to the stimulus. So they cannot do anything about that pain. They cannot actually move. So which part is damaged? So you have two types of neurons, a sensory neuron, well, three, but a sensory neuron, an interneuron, and a motor neuron. Okay, so where do we find, so a sensory neuron is going to receive the signal, that is the one that the receptor that enables you to feel the pain, but you cannot react to the stimulus, which means what type of nerve is going to be damaged?
So Talon in Galaxy J6 says motor neuron.
That is correct. And where in the diagram is motor neuron? Because remember, it's asking you for the letter.
Mshlongo says E, and Talon as well.
Yes, that is correct. Okay, so E is your motor neuron. And then give the letter of the effector. So the motor neuron goes to where? It goes to the effector. Okay.
Don said F.
Yes. Okay, so that would be your muscle at F. Okay, Nimisha, if you could please read for me.
Okay, so during a national census done in 2010 in a certain country, information was gathered on the number of people in the population who suffered from different eye diseases or disorders. The total population of this country was 142 million. So the results are shown in the table below. So on the left column, you have type of eye disease or disorder, and in the right column, you have the number of people in millions.
Okay, so and what's our first question?
So question 2.2.1, which eye disease or disorder is the least common in the population?
Okay, so it'll be the least number of millions of people. What's the answer?
Asante says macular degeneration. So least, remember, is less. So the most little, the smallest number, least.
Okay, yes, that is correct. Macular degeneration, okay, is the least common. Okay, then what percentage of the population is suffering from long-sightedness? Okay, so you've got 14.1 million. Okay, 14.1 million people are suffering from long-sightedness out of a total of all those people. Okay, so show all your working. What percentage of the population? So how are you going to work that out? You're going to add all of those numbers up, and then you're going to say 14.1 divided by the total of all of those people times by 100. Did everybody get that?
So the total population is 142 million. So ideally, when you add up all, it should give you 142.
Yes. Oh, sorry. Yes, it's given you that there on the top, 142 million. So you're going to say 14.1 divided by 142 million times by 100. That's going to give you the percentage of the population suffering from long-sightedness. Is everybody with me? Give us a thumbs up if you're with us.
So Nandi worked it out as 9.92%.
Very good. Thank you very much. Okay, now you are asked to draw a bar graph to represent the number of people in the population that suffer from cataracts, short-sightedness, and long-sightedness. Okay. So you're just going to have 3 bars on this bar graph, and you can label them. Okay, you can just put your categories on the x-axis. So it'll be cataracts, short-sightedness, and long-sightedness, a bar for each of those. Okay, and on your x-axis, you're going to write what? What is going to be the title of your x-axis? Type of eye disorder or eye disease. Okay, those are your categories. Then your dependent variable is going to be on your y-axis. So that would be number of people, and you must put the units. So that is now in millions. Okay, so it'll be number of people and millions in brackets for your y-axis. Okay, and then you can draw your 3 bars for cataracts, short-sightedness, and long-sightedness.
Jerusha, just
Yes.
a request if the answers will appear on the slides like the first session.
So I didn't put them because they were not appearing when I clicked. So I didn't put them up today. There's some up, but it's later on in the slideshow. So you have to just pay attention to what I say about it. So just scribble it down as I'm telling you the answers, but no, they're not up here for the first couple of slides, but I did manage to put some at the end for you, the long answers only. Okay, and in a bar graph, another point is that you must also still have a title of your bar graph. You can say figure 1, and you can say bar graph showing the number of people in the population that suffer from cataract, short-sightedness, and long-sightedness. So your title must be very descriptive. Do not say population versus eye disease. That's not a title. Okay, so you must be very specific, and it must be descriptive. Your title must say exactly what is being shown in that graph. Okay, and the same with a title for your diagram. It must tell you exactly what the person is looking at. Okay, Okay, 2.2.4, which eye disease or disorder in the table is A, the result of the lens becoming cloudy or opaque? So what's the word for cloudy or opaque? Lens that becomes cloudy or opaque?
They say cataracts. The answer.
Correct. Okay, that is correct. Which eye disease is found in people that are able to see nearby objects clearly while distant objects are blurred?
Short-sightedness, according to Tando. And Don said long-sightedness.
Oh, okay, so nearby objects are clear. That would be short-sightedness. Okay, distant objects are unclear. Okay, and it's the opposite way around for long-sightedness. So it's called short-sightedness because you can see something a short distance away. Okay, so that means you can see things close by, you can see things nearby clearly, but you can't see things far away. And the same for long-sightedness. You can see things far away, but you cannot see things close by. Okay, so short-sightedness, you can see short. You have sight for a short distance. Remember it that way. Okay, so short-sightedness, you have sight for a short distance. So that is clear. Long-sightedness, you have sight for a long distance. So you can see things far away. Okay, is that making sense? Does that help you to remember that? Students, you're not very responsive to me today.
I think they're tired.
You're sleepy. I know it's the evening, but I'm very glad that you guys are up after hours studying for your tests. Very good. Okay, let's move on.
Where's the mother? Hey, fuck. You want my, as it is.
Jerusha is not speaking as yet, so
I'm so
don't
sorry.
worry.
This PowerPoint is not behaving today. Can you see my screen?
Yes.
Okay. I'm just gonna leave it on this, okay? Because it's when I have it big, I'll put it up big for diagrams, okay? Can everybody see this? Let me pick this up. Okay, can you see this?
Yes.
It's clear enough. Okay, I'm leaving it on this because it's just being very problematic on slideshow. Okay, so the graph below shows the relationship between the levels of ADH in the blood and tubular reabsorption in the kidney. Okay, so we're talking about endocrine. Okay, so which structure in the body produces ADH?
Talon says thyroid gland. And Vanello says hypothalamus.
Okay. What do you think is the correct answer from those two? The thyroid gland produces thyroxine, okay? But ADH is antidiuretic hormone, which is produced by the hypothalamus. Okay. Now describe the relationship shown in the graph. What kind of a trend is that?
Directly proportional.
Correct. It's a directly proportional relationship. So when one increases, so does the other. So the tubular reabsorption increases as the level of ADH increases. Okay. So you must state that fully to get those 2 marks. Okay. Now for 3 marks, explain why the level of ADH at X probably indicates that the person is experiencing a cold day. So at X, it was cold. And how do we know this? Compared to, let's say, this point here. At 2.5, let's say. Explain why the level of ADH probably indicates that the person is experiencing a cold day. So I'll give you a clue. It's a low level of ADH. What does that mean? Why would that, what would that mean for, let's say, tubular reabsorption? If it's a low level of ADH, it means that there's going to be what? Read it off the graph. So what does that mean for tubular reabsorption? Is it low or high?
Low. Asanda and Santuri say low. Galaxy A3.
Okay, so low, it's a directly proportional relationship. So when ADH is low, so is tubular reabsorption. Okay, so tubular reabsorption means it's going to take back that water into the body. The kidneys will reabsorb the water. Okay, so the kidneys are going to now not absorb as much water because tubular reabsorption is low. Okay, so which means that the person will urinate more and the urine will be more dilute. Okay, so that's what happens on a cold day. Is everybody with me? Do you need me to repeat that?
And because generally on a cold day, you will visit the loo more often because of this low tubular reabsorption. And that's how your low levels of ADH is correlated to your low tubular reabsorption.
Okay, so your kidneys become less permeable to water, and that means it's going to be passed out via urine. Okay. On a cold day. Because on a cold day, you're not sweating, you're not getting rid of water. Your body has more water on a cold day. So more water means less ADH, and that means less tubular reabsorption. Okay. Okay, now the diagram below represents a possible path followed by an impulse when a person touches a hot plate. Okay. Name the path presented in the diagram. Well, you only know of one type of pathway. So what is this pathway? Students, any answers?
Don says a reflex action.
That is correct. Okay. Identify the type of neuron represented by B. What type of neuron is B?
Don says B is a motor neuron.
Correct. And a way to see which is which, in a motor neuron, your cell body is at one end. Okay, you can see a big cell body there with dendrites coming off of it. Okay, and then an axon. Okay, But excuse me. A sensory neuron has the cell body in the middle. Okay, so it's not at one end. Okay, so if you look at E, you can see that the cell body is in the middle. That's the difference between a sensory neuron and a motor neuron. Okay, so E, which is the answer for C, E is your sensory neuron. Okay, so what is C? What is the one in the middle there? The neuron in the middle.
Students are saying interneuron.
Correct. Okay. Give the letter only of the part that represents the receptor. So what is feeling the impulse? What is receiving the impulse?
F.
Correct. Correct. And what is the effector?
A.
Yes, that is correct. Asanda, do you have a question?
Okay, so Asanda's question is that Asanda does not understand the difference between a reflex arc and a reflex action when it comes to identifying it on a diagram.
Okay, it's the same thing. It's the same thing. A reflex, do you mean like a diagram of a spinal cord or nerves? Asanda? Can you just unmute and just answer me so that I know what your question is exactly?
Okay. Can you hear me?
Yes, young lady.
Thank you. When I see diagrams, I really don't know whether to say it's a reflex arc or a reflex action.
Yeah, it doesn't matter. It doesn't matter. You can say either or.
Okay.
Yeah. But you must be, if you see a sensory neuron, if you see a diagram like this where it's a sensory neuron and a motor neuron connected, then you must know, okay, it's a reflex action or it's a reflex arc, either or. If you see a diagram like this, the one with the spinal cord, it's also a reflex arc.
Thank you.
A reflex arc produces a reflex action. Are you with me?
Yes.
Okay. Okay. Let's move on. Give the letter and name of the region where the impulse is transmitted chemically. So transmitted means it's moving from one place to the next. Okay, so where is this impulse transmitted chemically?
John says D, the synapse.
Correct, okay. And a part that has an insulating function. What insulates?
Don says G, the myelin sheath.
Yes, that is correct. Okay. And the purpose of insulation is so that the signal gets transmitted faster. Okay. Remember, it's an electrical impulse, so there needs to be insulation. Okay. So name the part of the brain that receives impulses from parts A and B. Any answers?
Galaxy A3 says the cerebellum.
That is correct. What type of impulse is being transmitted? Students, what type of impulse is being transmitted by A and B? an impulse about what?
Don says balance.
Yes, that is correct. Okay. Okay. Movement along a plane. Okay. What is the name of the part of the brain that receives impulses from part C?
Don says cerebrum.
Yes, because it is picking up an impulse of sound. Okay. Name the receptor found in part C. What is the receptor called? I'll give you a clue. It's an organ. It's called an organ.
Don said the organ of Corti.
Yes, that is very good. Correct. Okay. Explain two ways in which part A in the diagram is structurally suited to maintain balance. Okay, so how is part A, those three canals, those three semicircular canals, how are those structures suited to maintain balance? For 4 marks. Any answers?
No answers yet.
Okay, so the semicircular canals are located or set in 3 different planes. So that means one is coming forward, one is going backwards, and one is diagonal. Okay, it's in 3 different planes, so it can detect movement in any direction, whether you're going forward, backwards, up, down, left, right, in any direction. The semicircular canals will be able to perceive that because they have cristae, okay, that will actually move to be able to interpret that movement that you are doing. Okay, so the semicircular canals contain endolymph, okay, and that is the liquid that actually moves to in response to how your body is moving. Okay, the semicircular canals also have what we call maculae, okay, and these maculae sense the change in the position of your head. Okay, so it affects your balance. Okay, and I've put in an answer for you. Okay, so the semicircular canals contain fluid or endolymph, which moves as when the person moves, and there are cristae or receptors present which convert a stimulus to an impulse. Okay, so the stimulus being movement, okay, converts it to an impulse, and those cristae are sensitive to movement of the fluid in them. Okay, and then lastly, the canals lie on 3 different planes to detect movement in any direction. Okay, if you're taking that down. Okay, you need to know how that works. How your body is able to maintain balance. Okay, is everybody fine with that? Okay. Identify the following parts in the diagram, and we can see a diagram of the eye. Okay, what is part B?
Don says the choroid.
No. Try again.
The C says sclera.
Yes. Okay. That is correct. And what is part C? What is part C?
I'm still getting sclera as the answer. Conjunctiva by Tassi. And Santuri is asking if it's the retina.
Sorry, I'm looking at the incorrect thing. Whoever said B was the choroid is correct, and C is the sclera. Okay, so B is your choroid, and so, yeah, the label for B is choroid, and the label for C is sclera. Okay. Explain the effect on a person's vision if part E is cut. Okay, what is that? What is part E?
So Seema says the optic nerve, and Dawn said the person will be blind in one eye.
Yes, that is correct. Okay, because the optic nerve is what transmits that impulse of light, that stimulus of light into an impulse and transmits it to the brain. Okay, so if it can't transmit to the brain, the person is going to not be able to see anything. Part D can be damaged by very bright light. Okay, describe how part A helps to protect part D in very bright light. Okay, so we're talking about pupillary mechanisms. Okay, so what happens in bright light? You can see this question has come up twice now. So we already gave this answer in a previous question. In bright light, these circular muscles of the iris contract and your radial muscles relax. Okay, so what does that do? That makes the pupil size decrease. Okay, it makes the pupil smaller, which allows less light to enter the eye. And that will protect the retina from bright light, from very bright light. Okay. Is everybody with me on that?
Yes.
Okay. In a condition called presbyopia, lenses lose their elasticity and therefore maintain a constant flat shape. Explain how this condition would affect a person's vision if the lens is constantly flat. Would it be able to bend the light or refract the light onto the retina?
No.
No, it wouldn't be. Okay, so explain how this condition would affect a person's vision. They would constantly be seeing something that is blur. Okay, but I have a nice long answer for you. Okay, what will happen is that the person will not be able to focus that image because the lens cannot undergo accommodation. The lens cannot bend, okay, because it's constantly flat. So accommodation will not occur. The refractive power of the lens is low, or the lens cannot become more convex or more curved. And the light rays are not refracted or bent such that it lands on the retina. Okay, so it will not be focused onto the retina. And that means you would not get a clear image. Therefore, the person cannot focus on objects that are closer than 6 meters because it cannot curve that lens to see things that are close by. The person can only focus on distant objects. Okay. Suggest the shape of the lens that may be prescribed by a doctor to correct this order. So we are trying to make a flat lens more convex or more rounded. So the doctor would prescribe a convex lens. Okay. Okay. Nimisha, if you can read the 3.2.
Okay. So 3.2, the endocrine system plays a role in helping a person to cope during a dangerous situation. 3.2.1, name the hormone that is secreted in the person's body in response to a dangerous situation.
Okay, what is the hormone that?
So Don said adrenaline.
Would be secreted. Yes, that is correct. Okay. So adrenaline is released in a dangerous or stressful situation. State 3 effects that the hormone in question 3.2.1, so adrenaline, has on the body. So what 3 effects does adrenaline have on the body? That is your fight or flight hormone.
So Don says a high heart rate.
Correct. What else?
A high blood pressure.
Yeah, I guess so.
Fast movement, according to Seema.
Sorry?
Seema said fast movement.
Fast movement.
Maybe more of a fast reaction.
Okay. Okay, but that is, yeah, that is what eventually happens. But what is making the movement fast?
So Santuri said there'll be an increase in metabolic activity.
Metabolic activity. Yes.
Seema also said there'll be an increase in hormone levels.
Okay, the answers that I'm getting are very vague. And I don't think you're going to get marks for this type of answers. This is what you need to learn off. Okay. Adrenaline prepares the body for crisis. Okay. It increases the heart rate. And if you increase your heart rate, you're also increasing your breathing rate. Okay. Adrenaline causes glucose levels in the blood to increase. Why does glucose levels need to increase in the blood? So that the glucose can go to the muscles and cause the muscles to contract, do whatever that the muscles being able to get that glucose is now what will allow it to move fast. Okay, so let's say you need to run away from something, you need the energy immediately to be able to run away. Okay, so that is how the hormone adrenaline will help you in a dangerous situation. Okay, but the role of the hormone is not to make you run away, it's to make glucose available in your bloodstream. Okay, do you see what I'm trying to say? That you need to tell me a biological or a molecular response rather than what we see on the outside. Okay, so glucose levels in the blood increase. That is the direct role of adrenaline. Okay, and then the tone of your skeletal muscles will also increase. So tone means you'll be able to have an effect. It'll be quite contract. It'll be more contracted. Okay, and ready for. It'll be in a good shape to be able to have an effect or cause a response, like running away. Okay, so you need to be able to give me direct effects of adrenaline, not vague things. Okay, because you won't get the marks. Okay, so at least remember these 4 points. increase heart rate, increase breathing rate, increase glucose levels in the blood, increase tone of skeletal muscles. Okay. Right. Then describe the homeostatic control of blood glucose levels in a person who consumed a drink with a large amount of sugar. So now we're talking about a negative feedback system when a negative feedback system of glucose of insulin when the glucose levels are high. Okay, so this is something that you must learn. You must be able to say this negative feedback system. Okay, so when your levels of glucose in the blood are high, what happens? Okay, I need an answer for 5 marks, please.
Glucagon gets released.
You're telling me the last point. What's the first thing that happens? How does glucagon get released?
By the alpha cells.
Okay, first, sorry, that is incorrect. We don't want glucagon being released. Your glucose levels are high, which means that you need to take some glucose out of the blood, which means you need to store some glucose. Okay, so what stores glucose? Glycogen. Okay, so we need increased glycogen levels. And how does that happen? Students, I need an answer. You need to know this. So when glucose levels in the blood are too high, the pancreas is going to be involved here. The pancreas will detect this, okay? And in the pancreas you have what type of cells?
Islets of Langerhans. It secretes more insulin, according to Mfanele.
That is correct, but now be more specific. I want you to be more specific. You're going to tell me beta cells of the islets of Langerhans. Okay, so the beta cells of the islets of Langerhans in the pancreas will now secrete the hormone. What hormone?
Insulin.
Very good. Okay, so insulin will be secreted. And then insulin now converts excess glucose, because remember we've got too much glucose in the blood. It will convert that extra glucose into glycogen. Okay. So
Santuri
insulin, yeah.
Santuri said the beta cells of islets of Langerhans in the pancreas.
Yes, that is what will secrete your insulin. And the insulin will convert excess glucose to glycogen. So insulin promotes the entry of glucose into the blood, into the body cells. Okay, so it will come out of the blood and be stored in the body cells as glycogen. Okay, and now because glucose is leaving the bloodstream and going into the cells, that is what's going to make your glucose levels in the blood decrease. So it will come back to normal level. Minenhle, do you have a question? Minenhle?
Minenhle, you are muted. Unmute your mic.
Is she typing something?
No.
Okay, I'm gonna move on. Okay, I've put an answer there for you. Okay, blood glucose levels rise above normal, the pancreas or the beta cells of the islets of Langerhans secrete insulin into the blood, which travels to the liver and muscle cells and stimulates them to absorb glucose from the blood and to convert excess glucose to glycogen. This now decreases the blood sugar levels back to normal. Okay. Okay. If you can read that top line for me, Minisha.
Minisha, you still have your question?
Yeah, it's okay now.
Okay.
Oh, okay.
You sure?
Yeah, I wanted 3.2.
3.2. I can tell you 3.2. 3.2.1 or 0.2.
3.2.2. 3.2.
Okay.
Yeah.
Okay.
The
The
enrollment,
state
yeah.
3. Sorry.
I may proceed.
You sure you got the answer?
Can you repeat me the endocrine system play a role helping a person cope during a dangerous situation?
Okay.
I want an
So
answer for
the hormone. Okay, so the question is name the hormone that is secreted in the person's body in response to a dangerous situation, and that hormone is adrenaline. Okay, and now 3 effects of adrenaline in the body. So direct effects of adrenaline would be to increase your heart rate, increase your breathing rate, increase your glucose levels in the blood, and increase the tone of skeletal muscle.
Okay.
Okay. And you got the last one? The answer is on the screen.
Let's say blood glucose level.
Yes, that's the answer for 3.3.
That's the answer for 3.3.
I have. Thanks.
Okay. Okay, so Minisha, if you can just read the top for me, please.
Okay, so an investigation was carried out to determine the influence of alcohol on the volume of urine produced. 12 healthy 23-year-old males of similar height and mass participated in the investigation. The investigation was conducted as follows. The men were divided into 2 groups of 6 each, Group A and Group B. The 2 groups ate the same food and did the same exercise for the 24 hours period before testing. Each group was given the following to drink after the 24-hour period. Group A, 1 liter of alcohol-free beer, beer that does not contain alcohol. Group B, 1 liter of alcoholic beer. The urine was collected from each man every hour. So assume that the volume of urine collected is equal to the volume of urine produced. The results of the investigation are shown in the table below.
Okay, so we can see that after 1 hour, the average volume of urine collected from Group A was 599, whereas that of Group B was 643. Okay, so there was always, after 1, 2, and 3 hours, there was always more urine produced by Group B than Group A. Okay, so state the dependent variable in this investigation. What is the dependent variable, the one we are measuring?
Urine.
Don't just say urine, tell me the full answer.
I think average value of urine collected.
In milliliters. Okay, that is the full correct answer. Okay. 2 planning steps the investigators had to take before the investigation could start. They like to ask this question about planning steps because they did so, they asked this even in the other questions we were looking at on Tuesday. 2 planning steps. So they're asking you to think about the method. They had to decide or select the participants, okay? They had to decide how much of alcohol they're going to give them. They had to decide how long they are going to run the experiment for, after how many hours. They had quite a few things to think about, okay? So that's just, you can think about any of those factors. I mean, you can state any of those factors. Okay, name 2 factors that need to remain constant other than the ones you already mentioned. Okay, so it must not be the same answer as B.
The amount of time.
Of what?
Time.
There's different times. You have to be specific.
Yeah. Tell me time of what?
Increase in sample size.
Sample size is correct, yes.
Asipa said the age of the males.
Yes, very good. But tell me, let's just go back to the time. Time of what?
Of hours.
Time of collections.
Yes, time of collection. Okay. So please be very specific. Don't just say time, you won't get the mark. You need to be very specific.
Because in the description, one is mentioned the 24-hour period following drinking, and the other one refers to hours of sampling. So you need to be specific to which hour you are referring to.
Okay.
Okay.
2 steps that the investigators took to ensure the reliability of the investigation. So how did they make sure that this investigation was reliable?
Some during the investigation.
Sorry?
I think some during the investigations.
What do you mean by that?
You mean having a control and a test group, Minisha?
I think I made a mistake.
No, you're sort of correct. Just explain that a bit more. So how do you increase the reliability of your investigation? Can you do an experiment once and then
by repeating the investigation?
Right. Okay. And what about the number of subjects? Okay, so they used 12 healthy 23-year-old males. They didn't just use 2. Okay, so they used 12, which is a good sample size. Okay, so they used a reliable sample size. Okay. What else? If you look at bullet point number 2, the 2 groups ate the same food and did the same exercise. Okay, they controlled those variables, which increases the reliability of the investigation. Okay, based on the results, explain how the intake of alcohol influences the secretion of ADH and consequently the volume of urine that is produced by the kidneys. Okay, so what is the influence of alcohol on secretion of ADH? Based on the results. So group A, the ones that had alcohol-free beer, produced less urine than group B, which had alcoholic beer. Okay, so alcohol does what to ADH?
Okay, think about the previous question with the graph with ADH and tubular reabsorption, and then think about your answer. So Asanda said it decreases ADH.
It decreases ADH.
And alcohol increases the urine amount, Asiba.
Okay, make that link more clearly to me, please. So alcohol affects ADH how? How does it influence the secretion of ADH? Does it increase or decrease it?
Breathing is increasing.
You think it's increasing? So the people in group B had increased ADH. That's not correct because they made more urine, which means that the water in their body was not reabsorbed. Okay, so remember ADH is directly proportional to tubular reabsorption. Okay, so if you have increased ADH, it means you have increased tubular reabsorption, which means that you have less urine. Okay, so alcohol actually decreases ADH. And if you have decreased ADH, you have decreased tubular reabsorption, which means you have increased volume of urine. Okay, so your kidneys are reabsorbing less urine, less water, which means you're gonna urinate more water out. Okay, I think I have an answer for you. Okay, alcohol inhibits or reduces the secretion of ADH, which causes the renal tubules or the distal convoluted tubules and collecting ducts to become less permeable to water. Water is less reabsorbed, or less water is reabsorbed rather, back into the blood, which means a larger volume of urine is produced. For 4 marks. Okay, do you have that?
Yes.
Okay. Okay, now identify, we can see the pictures of the eye there. Identify part A. Hello.
Don said desclera and some tutoring.
That is
It's the outermost covering.
Asanda said cornea, but cornea is not.
No, it's your conjunctiva. Okay.
Asima said conjunctiva now.
Yeah. And B?
Lens.
the lens.
Okay. And C?
The ciliary
Ciliary
body.
Yes, ciliary body. Okay. Identify the process in diagram 1. What's happening in diagram 1? The pupil is going from very big to very small. So what is that called?
Change shape, near and distance.
It's
vision.
called pupillary.
Pupillary.
Pupillary adjustment, you can say. Okay. Name the part of the eye that is responsible for the response in diagram 1.
Iris.
So what is making that pupil change size?
The iris.
Yes.
It's the iris.
Yes. State the consequence to the person's vision if the process in diagram 2 does not occur.
Too much of light will get into the eye.
Okay, first of all, tell me what's happening. What is happening in diagram 2?
The lens changed the
The
shape.
pupil is getting smaller.
Correct. No, in diagram 2, we've got the lens changing shape. Okay. State the consequence to the person's vision if the process in diagram 2 did not occur. So when the lens changes shape or becomes more convex or less convex, that is called accommodation. So if accommodation does not occur, that means the person's vision is going to be blurry. Okay, accommodation happens to make an object or an image clear by changing the shape of the lens. Okay, are you with me?
Yes.
Okay. Okay, if you can read that on the top there for me, please.
Okay, so a normal, healthy person was placed in a cold room. After 30 minutes, the thermal image shown below was produced. This thermal image indicates the temperature of different parts of the body. Lighter colors on the scan indicate temperatures lower than normal body temperature.
Okay, so you can see the outer part of the person's body is gray and the inner part is black. Okay, so the inner part was more hot than the outer part. As you can see, the hands and feet are white. Okay, which means that they were very cold. Okay, which color, black, gray, or white, represents the normal body temperature?
Black.
Black.
The black. Okay. State what occurred in each of the following parts in this person's skin during temperature regulation. So what happened to the blood vessels in the person's skin? Because it was very cold. So what happened to the blood vessels in cold weather? At cold temperatures, I mean. Do blood vessels dilate or contract in cold temperatures?
Can you write there for me?
They said contract.
Okay. Okay, so blood vessels contract. So that
Seema said,
yeah.
So it contracts and it diverts all the warm blood to the torso of the body.
Yes, okay. That is to keep the vital organs warm and functioning. Okay. So your blood vessels contract so that you don't have too much of blood. So you'll have too much of blood on the periphery. So it'll be less heat lost from the blood. Then your sweat glands, what happens to the sweat glands?
They become less active.
Yes, they become less active. You don't sweat in cold weather. Okay. Name the part of the brain that is responsible for thermoregulation.
Hypothalamus.
Yes.
Dawn said the cerebrum.
Okay, so which is it? Hypothalamus or cerebrum?
CP said hypothalamus. 3 for
Yes.
hypothalamus.
Okay, so hypothalamus controls your body temperature. That is correct. Okay. State one way in which the thermal image would differ if the person was placed in a hot room for 30 minutes. So what would the diagram look like? What color would it be?
White.
No.
It will be
Gray.
all black.
It will be full black, okay? Because lighter colors indicate temperatures lower than normal body temperature. So we won't have any light colors because it won't be cold. It'll be warm. So that would be all black or gray. Okay. Okay. I can see the graph below shows the speed at which impulses are transmitted along sensory neuron axons of increasing diameter when the axons are myelinated and unmyelinated. Okay, so myelinated means covered with a myelin sheath, and unmyelinated means there's no sheath present. Okay, so a comparison of the speed of an impulse in myelinated and unmyelinated neurons, sensory neurons. And the diameter of those neurons are increasing. Okay, so it's like a thicker or thinner rope. Okay, the dotted line in the diagram is myelinated. Okay, so describe the direction of the impulse within a neuron. Which direction does an impulse travel? From where to where? From the dendrites to the cell body. Okay, and the axon transmits signals away from the cell body to the effector.
Can you write down for me that one?
Okay, I'll repeat it. The direction of an impulse is the signal is received at the dendrites and transmitted towards the cell body. And the cell body now transmits that signal away through the axon towards the in this case, let's say the spinal cord. So an axon transmits signals away from the cell body. Okay, give the diameter in micrometers, the diameter range when the speed of the impulse is faster in unmyelinated axons than in myelinated axons. So where is the solid line higher than the dotted line? Between 0 and 1 diameter, 0 and 1 micrometer diameter. Okay. Are you with me?
Yes.
Yes.
Okay, describe the relationship between axon diameter and the speed of the impulse in myelinated axons. So we're only looking at the dotted line. So as the diameter increases, the speed also increases. Okay. It is a directly proportional relationship. Okay. Use evidence from the graph to explain the effect of multiple sclerosis on a sufferer whose motor neuron axons are greater than 1 micrometer. Okay, so we're looking at which line? What is multiple sclerosis? What happens in multiple sclerosis? Students, any answers?
No.
Okay, so multiple sclerosis is when that myelin sheath starts to get destroyed. Okay? Yes, the immune system damages that myelin sheath. Okay, so we are looking at the solid line. We're looking at nerves that are unmyelinated, neurons that are unmyelinated. Okay, so use the evidence from the graph to explain the effect of multiple sclerosis on a sufferer whose motor neuron axons are greater than 1 micrometer. Okay, so we're looking at unmyelinated because it's a patient with multiple sclerosis, and then their axons are greater than 1 micrometer. Okay, so what happens? What happens to the speed of that impulse? It increases.
It increases.
Very gradually. Okay, there is not a very big difference between a 4 micrometer axon compared to a 1 micrometer axon. Okay.
Yes.
Oh, sorry. I thought I had an answer there. Okay, so the speed of the impulse increases very gradually and is much, let's say, significantly lower. The speed is significantly lower than that of myelinated neurons. Okay. Okay, if you can read that, please, for me.
Okay, so 2 types of hearing loss occurs in humans. Conductive hearing loss occurs when sound vibrations cannot be conducted through the outer and middle ear. Sensory neural hearing loss occurs when sound waves in the inner ear are not converted into nerve impulses, or when the impulses cannot be transmitted to the brain. The diagram below represents the human ear. So question 2.4.1, give the letter and name of the part that A transmits impulses to the brain.
Auditory nerves.
Yes, the auditory nerve. Where is that? What's the letter? It's asking for the letter and the name.
Letter F.
Okay. Allows pressure to equalize between the outer and middle ear. What part?
It's parts.
Part H, round window.
Yes.
No.
Part H.
CP says B. What is B?
Part B is
Tympanic membrane. No.
I think it is D.
Yes, sorry. I
It
think
is
it's
D.
D is the semicircular canals. Okay, so the answer is G, the eustachian tube. That actually allows you to equalize pressure between the outer and inner ear. Okay. Okay, give the letter of 2 structures in the diagram of the ear that, when damaged, would result in the following. Conductive hearing loss. Okay, so when the sound vibrations cannot be conducted through the outer and middle ear. So what 2 labels do you see there? In the middle ear. That
One said B and C.
occurs when sound vibrations cannot be conducted through the outer and middle ear. Okay, so B and C. Okay, so the tympanic membrane has to vibrate, and that vibrates those three ossicles, which is C. Okay, so if those two things are not functioning, then you will get conductive hearing loss. Okay, what two structures would result in sensory neural hearing loss? That is when sound waves in the inner ear are not converted into nerve impulses, or when the impulses cannot be transmitted to the brain. So that means what transmits the nerve impulse? We said it is F. Okay, so the auditory nerve, and what transmits those signals? The organ of Corti. So we've got E and F. Okay. Middle ear infections are common cause of hearing loss. State one way in which middle ear infections are treated. What's one way that middle ear infections are treated?
Grommets are inserted into the tympanic membrane.
Punnett said a hearing aid.
Okay, grommets are inserted, that is correct. A middle ear infection, no. You wouldn't have a middle ear, you wouldn't have a hearing aid for a middle ear infection. Okay, a hearing aid only helps for hearing loss. Okay. Name the part of the ear where earwax is produced.
Ear canal.
Correct. Explain why hearing loss due to earwax is usually temporary. Why is it temporary? Because you can actually remove earwax and then your hearing is restored. Okay.
Jerusha?
Yes.
Tessie had a request that if you could just go over monohybrid and dihybrid cross for Tessie, because they're writing a genetics test tomorrow. If you could just go over the theory.
Yes, that is a good question for genetics one.
Sorry, please can you repeat that? What would you like me to go over?
Mono.
Genetic and like mono, like she said, as
Monohybrid cross and dihybrid.
Yes.
Yes.
Okay, monohybrid cross is what?
Is a one and the
One traits only.
Yes, that's correct. Okay, and a dihybrid cross is two traits. Okay. Okay, let me just get my notes quickly.
on that time they just died.
Sorry?
On that time did you start the class?
At 6. At 6 o'clock. Okay, so a monohybrid cross, okay, is when you're just crossing one trait. So what you're going to do is you always need to write the following things. You must always write this. The phenotype of the parents. So for example, tall and short. Okay. Then the genotype of the parents for that trait. Okay. So you need to know what's dominant and what's recessive. Okay. Then
Meiosis.
okay, there you go. Dr. Singh has put up a slide for you. Can you see that? It's a little bit small, Minisha. If you can just make it a bit bigger. Can you do that? Yes, that's better. Okay, so you will end up with, okay, so as I said now before, when you are doing any cross, the first thing you write is your phenotype, okay, then your genotype, your phenotype of the parents, then your genotype of the parents, then the gametes, okay? So in this case, the gametes are going to be big T and small t, okay?
Must we ring it?
Sorry?
Must we ring the T and the small letter T?
You don't need to circle it. You just need to write big T and small t there. Okay, those are your gametes. Then what happens when those two gametes are fertilized? Then now that's when you do your Punnett Square. Okay, so that's what's shown in that diagram there. Okay, so you got your two gametes, big T and small t, and then your after fertilization, you're gonna get big T, small t. Okay.
F1.
Then and that's your Punnett Square. Then you're going to interpret that Punnett Square. You're going to say, now in the F1 generation, I've got what's the genotypes and what's the phenotypes of the offspring. Okay, so in this case, you've got big T, small t. You've got 4 big T, small t. So that's your genotype. And what's your phenotype? You're going to say all, in this case, all of the offspring are tall, because tall was the dominant trait over short. Okay. Now in a dihybrid cross, you've got two traits being crossed. Okay. So what you're going to do is, it's the same exact thing. You're just going to put two different letters next to each other. Okay. Okay, so you're going to have same thing. The phenotype of the parents, they can be, so it'll be two different traits that you're writing there, two traits that you're showing. Let's say height and weight. Okay, so you've got a tall, skinny person crossed with a short, fat person. Sorry, this is a bad example. I can't see. Is that an R? R and a Y.
R and a Y, yeah. You want me to find another one? I can find a dihybrid one.
No, can you think of a better example?
Okay, so let's say this dihybrid cross is the color of flower, which is capital R for red, and the small r is for yellow. Then the Y's is for the color of the leaves. Capital Y is green, while a small y is brown. And then if you have, so in the terms of this dihybrid cross, you're having heterozygous for both genes being crossed for the parents. So of course, for each one, your cross is going to be, let me type the R, capital R, small r, capital Y, small. So this is your alleles for your cross. So we are saying here that red is dominant over your pink, and for your leaves, we're saying that green is dominant over brown. Just 1 second for me, let me close this box. So we need to generate the gametes. So when you do for a dihybrid cross, you need to pull out gametes for both of your traits. So you'll go capital R, capital Y, you put that down in your Punnett square. Then you do a capital R with a small y, you put it in the Punnett square. Then now we're going to do our recessive trait for pink. So it's small r with capital Y for green, you put that in the Punnett square. Then you put your both recessives, which is R and Y, you put that in this Punnett square. So we're saying two heterozygous parents are crossing. So of course, on either side, the one side is your maternal gametes, and on the top is your paternal. So you're going to have the same gametes. So like how you do for a monohybrid cross, where we're only looking at a single trait, the same rules apply for the cross. So you first will do for your flower color. So you're going to cross, you see that the first gamete for the maternal and the first gamete for the paternal, you're going to get two capital R's, or your dominant for red. You're going to cross again for the Y's, you're going to get two dominant for green. Then you're going to move along the gametes from the first row with the different gametes from the paternal. And you're going to have different variations. So you'll do this across for all four variations of the gametes. And you will now create different genotypes and phenotypes. Now, generally in a heterozygous cross, you will have all four variations of the phenotype showing. So you're going to have a red and a green, red flowers with green, then you're going to have red flowers with brown leaves, then you're going to have pink flowers with green leaves, then you're going to have pink flowers with your recessive brown leaf. So generally with the recessive in your Punnett square for a dihybrid cross, you usually have one, which is homozygous recessive for both traits. Right, and so generally when I do a dihybrid cross and when I have to do the genotypes and the phenotypes, the thing what I used to do when I used to do these crosses for tests and for exams, I used to use different color pens or shapes. And I used to write the first genotype. I will use maybe green. I'll put a green dot for all those that have all that is homozygous dominant. Then I'll go on to the heterozygous for the dominant and carry on with a different color. So then when you are writing down the genotype and the phenotypes, it's easier for you to count. So that's the trick I used to do for a dihybrid cross. I don't know if you have any other tips, Jerusha?
Yeah, that's a good one. You can use also like a highlighter. Just once you've counted them, just strike them off. So you shouldn't have more than 16. Okay, so you know when you're writing your phenotype of the offspring, make sure that you've got 16 offspring. Then when you're writing your genotype, 16 of them. Okay, so you know you've counted all. Okay, and also you can learn off what the offspring would be if you have a cross of two heterozygous parents. What will you get if you cross two homozygous parents? Okay, so if you can find those answers, then you know it'll be easier for you in the test.
And usually if it's a normal dihybrid cross, your phenotype is always the phenotype, right? 9:3:3:1. Or is the genotype? Did I do that wrong? Did I type the wrong thing here?
It's your phenotype, yeah.
Phenotype.
Yeah. Okay.
And also, if another trick that I always tell students as well to know that you did your heterozygous dihybrid cross correctly, let me just use a color. So you'll see in a diagonal, you're going to have your heterozygous across the Punnett. So you'll know that you did it right. If you have this diagonal of heterozygous for dominant, so you have big R, big Y, small y, you have done the cross correctly. So you knew you did your cross correctly, and you can now go on to counting your genotype and phenotype.
That's even for the dihybrid cross.
That's for, yes, that's for dihybrid.
Okay.
Okay, are there any other questions on monohybrid and dihybrid? Okay, Nandi has a question in the chat for an example of how you identify the independent and dependent variable. Okay, so
I'll stop share now.
The independent variable is the write it down like this. Independent is the effect of, whereas the dependent is the effect on. Okay, so the effect of temperature on sweat production. The effect of temperature on sweat production. So the independent variable is your sweat. The independent variable is your temperature, and the dependent variable is your sweat, amount of sweat. Okay, so independent variable is the one that you are manipulating. It's your treatments. Okay, it's the thing that you are controlling, and your dependent variable is the one you are measuring now in response to the independent variable. Okay, so the dependent variable is based on your independent variable.
Okay, thank you.
Okay. Any other questions, guys? All the best for your genetics test tomorrow. And I will see you on Saturday at 9 a.m. to 11 a.m. where we're going to be doing the genetics. Sorry, we're going to be doing DNA and meiosis. DNA and meiosis on Saturday morning, okay? And we're going to be doing genetics on Saturday evening. Okay, so 9 o'clock on Saturday morning. 9 to 11 and then 6 to 8.
Okay.
Okay, I'll see you all on Saturday. Please do come and tell your friends. If you found this helpful, tell your friends that you found this helpful and get them to join as well. Okay. Please don't forget to register. Click on that link and register if you haven't already done so.
Okay, so and this one I pasted the website link. Let me just paste it again. So at the moment, when I check the website, it's not there. So I'm going to query that for you so that it should be uploaded for you. I'll query with Ashwin about it. But it will be uploaded on that link there, and everyone clicked on the register link. One more time, the register link for those of you who haven't. Otherwise, everyone have a good evening, and we will see you Saturday.
Thank you, everybody. Good night. Thank you. You may log off. Bye.
Bye.